a valve that is called ileocecal valve, where the small intestine connects with the large intestine. You're going to find out in Sherwood that when pressure builds up on this side, or when chemicals that are released and hormones released from the small intestine reach this valve to tell it to open, then it will open. Otherwise, the smooth muscle will stay closed. The colon is full of bacteria which help to digest things, sometimes make problems for you, like lactose intolerance, and you get all this gas when you drink milk. A lot of people have that problem. But they also help to create vitamin K for our clotting system. So they're important to have. It's important to have the bacteria in the colon, but you don't want them to get into the small intestine. There are some bacteria in the small intestine, but most of them are in the large intestine, and that's what prevents the majority of the bacteria from getting into the small intestine. So this valve is actually a sphincter, a smooth muscle. It will allow a watery mass to come into the colon. And the cecum, which is the first part of the large intestine, is actually much more dilated than the artist has illustrated here. Because of this large watery mass coming in, the chamber, the wall is rather distended. And then it does narrow as it goes up to the flexure here. So this is an ascending colon. Specific name for the first part is the cecum. Another structure that is associated with the cecum is the vermiform appendix. Vermiform just means in the shape of a worm, and it does look like a worm. It generally is not hanging free like the artist illustrated, but it's held up, it's bent backwards and held up against the posterior wall of the cecum by some adventitia or some serosa. And that is actually helpful in that watery mass and bacteria-laden stuff doesn't get shoved into the appendix very much. Gravity forces water and heavy things downwards. And if you're constipated, if there's pressure building up because you can't get old stuff out, then you do get mass pushed into the appendix. If it's free, then there's a greater chance that you're going to get a lot of stuff into the appendix. When you look at the appendix histologically, it's full of lymphatic nodules. That tells you that there's a lot of bacteria that gets into it. And the immune system is there waiting for that bacteria and its antigens to arrive. So appendix, there are people who don't have their appendix there, they have their appendix somewhere else. And there are people who have multiple appendices, but usually the main one is they're associated with the cecum, and therefore it creates the greatest problem with that watery mass potentially getting into it. Watery mass full of bacteria. Ascending colon bends here right below the liver. So it's on the right side. It's called the right colic flexure, meaning it's flexing. The angle is becoming greater. It's becoming less. And it also is called the hepatic flexure because it's occurring right under the liver. Either term is perfectly acceptable, can be used interchangeably. The colon then goes across through the abdominal cavity. It's kind of hanging free. So it's going to be supported by mesentery that's going to come out and wrap around it and keep it attached to the posterior abdominal wall. And then it's going to flex again, and that's on the left side, so left colic flexure, also called the splenic flexure because the spleen is located in that area. The descending colon, if you look at a real descending colon, it looks like somebody took it and kind of stretched it and made it very long and narrow, and it's not going to be as wide as the ascending colon. And this makes perfect sense because the job of the colon is to dehydrate what comes into it, so take volume back, concentrate it, and then get a smaller mass as you're going through the colon. Some texts say that the descending colon stores fecal matter. No. The transverse colon is going to store the majority of the fecal matter, and when it gets into the descending colon, there's a massive wave. It's called mass peristalsis. So it gets into the descending colon, it's going to go right through, hit the sigmoid, and then into the rectum. When the rectum distends, then you're going to get a reflex contracture, well, reflex first relaxation of the smooth muscle sphincter, and then reflex contraction of the smooth muscle to push out that fecal matter, and that's called defecation. You take and measure the colon, it's about 5 feet in length, and the average diameter is around 3 inches. So the ascending is wider, the descending is narrower, and the transverse is kind of in between the two diameters. When you cut open the large intestine, you can see that there are bands on the inside, and those correspond to the bands of smooth muscle on the outside. In the esophagus, in the stomach, in the small intestine, the smooth muscle is all the way around the whole structure, but in the colon, this muscularis externus has been compressed into 3 bands that run longitudinally along the large intestine. They are called tinea coli. So here's one, and that's continuing as that band. Here's another one, and that's continuing down on the back side of the ascending and descending colon. And then there's another one on the back side that you can't see at all. So there's 3 of these bands condensed and running longitudinally. The circular smooth muscle is still there, and it's also been kind of compressed into little sections. And this, then the longitudinal muscle will dip down into the circular muscle in these areas and form these little pouches. The pouches are called haustra. The job of haustra are to kind of mix and turn and move things backwards and forwards, and what they're doing is mixing up the contents so that the watery contents get to come in contact with the surface epithelium so water can be reabsorbed. So if you didn't mix and turn it and cut it up and segment it and have it going in all directions, you'd have a dehydrated mass on the outside but a watery mass on the inside. So with all this mixing and turning, going back and forth, you get the watery stuff to the surface, and eventually everything is the same consistency. The intestine, large intestine, is lined with huge numbers of goblet cells. So you're dehydrating this waste product. You better have it slippery enough to get on through and out. So as you reabsorb the water, you leave a little bit there, but you coat it with mucus and make it slippery enough so that the smooth muscle contractions can push it on out. And elimination occurring. These omental appendages, those are oftentimes called epi, meaning on top of or upon cloaca. Sometimes they cause problems, but most of the time they're just little fat nodules that kind of hang on to the surface of the colon. This here is a large mass of fat, but that is actually part of the greater omentum that's just been cut away so you can see the surface of the colon underneath. So if you look over here, it says greater omentum cut. So greater omentum is actually been removed in that area, from that area. So here's a view of the ileocecal valve. You can see it's quite puckered. That's due to the smooth muscle that is in that area. You look, they kind of retracted it out so you can't see, you don't see too much of the ascending colon, but this is a little bit wider in diameter. You can see it's sort of tapering in as it's going upwards. And then vermiform appendix. I use that term, that's what I was taught. It's the one that looks like the worm. You can just say appendix, that's fine. The appendix is usually going to have the mesentery holding it up on the backside of the cecum. And so there's less chance for the fecal matter and bacteria to get into the appendix. The appendix is about 9 centimeters in length, so maybe a couple, 1.5 or so in length. The other things about the colon, they do not have villi, so less surface area. We're going to see in the physiology text, the surface area in the small intestine is there to absorb all the nutrients, but also to absorb the majority of the water that leaves your body to go into your digestive tract. That's a significant amount of water. It's about 7 liters of water goes into your digestive tract every single day. Your blood volume, if you're a guy, only 5 liters. So you can't ever afford to lose that 7 liters. Plus, you want your exhaling water, you have water leaving your skin, you have urine water going out. So you need some extra water to come back in. And the small intestine is going to absorb 9 liters. 7 liters goes in, 9 liters is reabsorbed, and you're left with about 500 mLs of water that gets into the large intestine. So when you get to the large intestine, no nutrients are reabsorbed. Don't need surface area for that. The water that's going to be reabsorbed is down to ½ a liter, and you're not going to reabsorb all of that. You're going to leave about 150 mLs around. So you don't need those villi. You don't need the microvilli. You're not doing very much there in the colon. I'm not making light of what the colon does, but it's not doing as much. It's not as active as the small intestine is. But it's going to have a huge number of goblet cells, because when it takes that last 350 mLs of water out of the remnants of digestion, you got a pretty chunky piece of stuff going through, and it's got to be slippery enough to get through the rest of the colon. Intestinal crypts are little glands at the base of invaginations in the surface of the colic mucosa. And they'll have cells down there that will be stem cells that will replenish the surface cells. They'll have cells that will release some antibactericidal enzymes enzymes to help to prevent bacteria from getting through the colon into the vascular system or lymphatic system. And they'll have some other secretions that are there. So these crypts are similar to what is happening in the small intestine. Again, because of the large bacterial colony in the small intestine, the large intestine, you're going to have a lot of lymphatic nodules in the wall of the colon. And that's because sometimes antigen and sometimes bacteria do get through the barriers, and you need to have your lymphatic cells, your immune system cells there to prepared to fight the bacteria before they get any further into your body. Rectum, there's a smooth muscle sphincter on the inside, that's your internal anal sphincter, and there's a skeletal muscle sphincter on the outside. That one's under your conscious control, but the internal one is not. When the rectum gets about 25% full, that internal sphincter relaxes. When it relaxes, it fires nerves to your brain, and your brain sends back nerves to the wall of the rectum to contract and push the fecal matter on out. So potty training teaches you how to contract this external anal sphincter to prevent inappropriate discharge of your fecal matter. So until you have control over those external sphincter muscles, you put something in the front and it comes out the backside. Liver, we're getting into accessory organs now. So we've got things that are growing out of the GI tract. We'll go to the duodenum. We've got two things growing out of the duodenum, liver and pancreas. The liver is a really busy organ. It does a lot of things for you. It's for digestive and metabolic functions. It's helping you with growth. You remember back last semester, but if you weren't in 301, then you don't remember that, but you'll get it in 301 if you take it eventually. Growth hormone is not able to do anything without getting the somatomedins from the liver. The first thing growth hormone does when it's released in the blood, go through the liver, pick up the somatomedins, and then together they go out to have their action in the peripheral tissue. So somatomedins are permissive. They allow growth hormone to have its action. The liver is going to make bile for you. We're going to learn what bile is in Sherwood. And it's kind of complex. You need cholesterol for it. You need fats for it. You need some salts. So we're going to learn about bile in chapter 16. The liver will also, if we have excess blood glucose, it can store about 150 grams of glucose as glycogen. And it also will store excess lipid. The liver uses a lot of lipid. It will make cholesterol for you out of lipid. It uses it to make your lipoproteins for transport of cholesterol out to your tissue. So it needs it needs lipid, so it stores lipid. It also can use lipid and chop it up and make new glucose out of it for you. So you can release the glucose from the glycogen, chop that up, glucose goes out immediately. You can get lipid from your adipose, lipid from your liver, and make new glucose out of that, and then keep your blood glucose in the correct range. So it is very important for maintaining normal blood glucose levels. If glucose is too high, takes glucose in, stores it as glycogen. If glucose is too low, chops up the glycogen, releases the glycogen out. It also helps to maintain amino acid concentrations in the blood and fatty acid concentrations in the blood. It will take amino acids and it will convert those into blood glucose. And it will take lipids and convert that into blood glucose. So it's taking different types of nutrients and making glucose out of them. It will synthesize cholesterol and then bind it to a protein that's your ApoB and transport it out to your cells where it will be utilized either to make steroid hormones, put it into the plasma membrane, or whatever the cells need the cholesterol for. So any source of cholesterol, the liver will utilize that. If it's in your diet, it will take the cholesterol from your animal consumption and utilize that for your own body's cholesterol levels. But if you are not eating animal products, it always amazes me. You go and you see a peanut butter jar or something like some vinegar and it's made of plants and it says cholesterol free. It's like, duh, yeah. And you get cholesterol only from animal products. You don't get it from plants. So anything that's made out of plants has to be cholesterol free. But the marketing, they'll use that to push a button. Oh, it's cholesterol free. But canola oil, any oil, any plant-based lipid, your liver will take it and make cholesterol out of it. And if you have inherited the wrong genetics, your liver goes crazy when it doesn't get dietary cholesterol. And it overproduces cholesterol. So there have been cases, 19-year-old vegan, dead. Why? Look, atherosclerotic, heart disease. But he's a vegan. Yeah, he's a vegan, but he inherited the wrong genetics. How do you know if you inherit the wrong genetics? You don't know. So better to get a little bit of dietary cholesterol than to end up dying young from atherosclerotic heart disease and not knowing what killed you because it's a silent killer, that's what they say. Anyway, so inactivating toxins. You drink alcohol, it will detoxify. You take in medication, it detoxifies the medication. You take any other illicit kind of drugs, it detoxifies you. You eat a salad and it's got pesticides on it, it detoxifies that. So your liver is there sucking up all these things that shouldn't be in your body. It considers medications and drugs and alcohol, things that are not supposed to be in your body. So it will take it, and some of the metabolites are stored in the liver. And if you eat liver, you are consuming that animal's toxins. Isn't that wonderful? I love this because our nutritionist kept saying, eat liver, eat liver, eat liver. And I said, I hate liver, I hate liver, I hate liver. And then I found out, oh, it stores all the toxins. Yeah, I don't have to eat liver because it stores all the toxins. So if you're going to eat liver, get the youngest animal's liver as possible, exposed to the least amount of toxins. But when you consume liver, you're consuming that animal's toxins. That's why it tastes so bad? Maybe so. It's pushed on you because it has a lot of iron. Why? Because it has a lot of blood in it. So it's a good source of iron, but it's an equally good source of toxins. So better to get your iron from something else, I think. I prefer something else. Because it stores lipids, anything that is soluble in lipids is going to be there in high concentration. So fat-soluble vitamins, A, E, B, and K, they're going to be there in the liver, in the fat. Other things things the liver does for you is make plasma proteins that keeps your fluid in your vascular system, so it doesn't leak out into your tissues and cause edema. It makes your clotting factors. That's the importance of vitamin K being in the liver, that you can make some of your clotting factors with that. It will inactivate the hormone angiotensinogen, which we'll talk about more when we get to the renal system. Again, we sort of brushed over it many times, but angiotensinogen is the precursor to angiotensin, which is releasing the ADH from your posterior pituitary and aldosterone from your adrenal cortex, and also is a vasoconstrictor. So the precursor, there's new stuff always coming out, so you got to get rid of the old stuff. So it inactivates the unused angiotensinogen. It has Kupffer cells, which are probably in the macrophage cell line, although other people will say, no, they're different. But they're phagocytes, so they're like macrophages. They hang out in the liver, and they will look for pathogen that's coming in through the blood. Since at least half of the blood is coming directly from your intestine, there's a chance that bacteria will have come through with the absorbed material and will be coming to the liver. So this blood passing through the liver, the first chance with that first pass for your immune system to get out any bacteria or pathogen in that blood. It's a good source of blood. They say it stores blood, but it doesn't open the door and shove blood in and store it. It's just that there's so many capillaries in the liver, all these liver sinusoids with blood running through them. It's just full of blood. So when if you cut the liver, a lot of blood's going to come out of the liver. It's because of the large vascular system that's in the liver. If the liver is compressed, it has a capsule on it, and that capsule can explode and rupture these blood vessels. And if you remember back to the fetal circulation, you had that ductus venosus that went through the liver when you were in the uterus, but then when you were born, it shut down and became a ligament. And that ligament, if the liver accelerates, it acts just like a piece of dental floss or something. You slice your liver, you get huge intra-abdominal bleeding from an injury like that. So it's important because it has a lot of blood in it. You don't want to rupture the liver or lacerate your liver. It's also sitting right there by your free-floating ribs, so you don't want to punch a rib into it either. It will break down circulating hormones like insulin and your neurotransmitter, epinephrine, and well, it's a hormone, but acting like a neurotransmitter. And it will also take old immunoglobulins, any old proteins, out of the circulation. And it will, as part of the detoxification, elimination of toxins, that will include any lipid-soluble drugs that you might have consumed. So liver is up on the right side in most people. Some people have their liver on the left, but the majority will have it on the right. Just depends on how things rotate in utero. The liver will go up to the right, and with that rotation, it's going to pull the duodenum around behind the stomach, and the large intestine will come with the liver, so you get the ascending and then the transverse and descending colon. So all that positioning is happening when that liver rotates up to the right and the duodenum and the pancreas go around to the back. Then everything else is distributed as it's going to be positioned. The liver has two main lobes, huge right lobe, medium-sized left lobe, and then underneath you'll see two smaller lobes, a four-sided lobe, so called quadrate, and then one that looks kind of pointed at the end, so they call that the caudate, meaning it looks like it has a little tail on it. So you can't see the quadrate and the caudate lobe when you look at the liver in that position. When you turn it upside down, you can find that. The quadrate lobe will be by the gallbladder, so that will always be your way to find your quadrate lobe. When we study liver, we study pig liver first, because pigs are very organized in their lobular structures. Everything is nice and hexagonal in the pig liver. When you get to human liver, everything is kind of skewed and squished looking. So in order to get a good feel for how things are organized, you look at the pig liver, which has a nice hexagonal arrangement, central veins nicely put in the center, and then hepatocytes that are sort of arranged like spokes on a bicycle. So the axle is your central vein, and then the spokes are the hepatocytes going out towards the edges of the lobule. We've got a hexagonal lobule and another one there with the blood vessels in it. The blood vessels have been pulled out so you can see the space in between these spoke-like arranged, linearly arranged hepatocytes. So these cuboidal cells that are stacked on each other and then lined up in radial arrangement towards the periphery, those are your hepatocytes. They're going to be looking at the port of blood. This is a branch of the hepatic portal vein. They're going to look at the port of blood for nutrients, take the nutrients that they want from that port of blood, and then they're going to look at, there's going to be between the hepatocytes and the blood vessels, Kupffer cells that will look for pathogen that might be there. Blood that is in the hepatic artery is going to contain the fats, the majority of the fats that your intestines absorb. So that is the source of the lipid that the hepatocytes will take out and then utilize or store for later use. The hepatocytes will take out things necessary to make bile, and then they will make the bile and then secrete it in between adjacent hepatocytes into small little canals called canaliculi. Same name as the bone, like the structures in the bone, that in the osteons. So it just means a small little narrow canal. The bile then will go into larger and larger ducts. The ducts are always very easily identifiable because they have a nice clear lumen and then very cuboidal cells with very nice plump centrally located nuclei. So the duct is easy to identify. The arteries are fairly easy to identify because they stay round. They have a lot of smooth muscle around them. And the veins, they'll have all different kinds of shapes, less smooth muscle, all kind of skewed and flattened out. But they'll always be together. In the pig liver, they're very neatly arranged at each corner of this hexagon. And that's what they're shown here. This is referred to as a portal triad. So three things together. You've got the hepatic artery branch of that, a branch of the hepatic portal vein, and then a branch of the bile system. And at each corner, In the human, you might have something that looks like a corner, but no portal triad there. So it's not as neatly arranged. But once you get the system down and you know when you see a vein, then you look closely, you're going to be able to locate an artery and a bile duct. You find a bile duct, look closely, you're going to find a branch of the portal vein and the artery. They're going to be there, but they're not going to be as nicely arranged as in the pig liver. Everything that goes from the portal vein or the hepatic artery through these sinusoids, what isn't taken out by the hepatocytes will be collected in the central vein, and the central veins will join together and form the hepatic vein that will take all of this blood to the vena cava. Do not get the central vein confused with the hepatic portal vein. Do not get the hepatic portal vein confused with the hepatic vein. They're all different structures. The hepatic portal vein brings blood to the liver. When the hepatocytes are through with the blood and have taken what they need out, or what shouldn't be there out by the Kupffer cells or the hepatocytes detoxifying you, then the rest will go into the central vein, and it will be at least somewhat cleaner to go into the systemic circulation. So a little bit enlarged, but a little bit wrong too. The Kupffer cells are not in the sinusoids. The artist drew them in the sinusoid. They're not. They're outside the sinusoid. They're between the hepatocyte and the endothelial cells of the sinusoid. So they're in that space there. And they even name that space, the space of D-I-S-S-E. So they're out here, but these sinusoids, they're not solid like the artist drew. They have big gaps between their endothelial cells. So very easy for this Kupffer cell to stick its little foot processes out into the sinusoid and find the pathogen or the antigen that's not supposed to be there. So they work in conjunction with the sinusoid, but they're not in the sinusoid. The bile that is collected in all the small bile ducts will go into bigger and bigger and bigger ducts. And then you'll have a main duct that comes from the right lobe, and that will be the right hepatic bile duct. There'll be one from the left, and there'll be smaller ones from the quadrate and caudate lobe. And most textbooks kind of ignore that because they're so small lobes. Those bile ducts will come together and form one bile duct. That joining together forms a common hepatic duct. The common hepatic duct is heading the bile towards the duodenum. But there's a sphincter at the duodenum that isn't always open. So if the bile is not getting out into the duodenum, then the fluid pressure will build back up through the duct system, and the bile will go into the gallbladder. And the gallbladder has epithelium that's designed to reabsorb huge amounts of water and concentrate that bile there. This duct here is called the cystic duct. The gallbladder is a bile-filled sac, a fluid-filled sac that is called a cyst. So this is then okay to call it the cystic duct. Bile is green. Gallbladder will have a greenish color to it. So will the ducts. So up here where the left hepatic and right hepatic are joining, there will be smaller ones from the other lobes, but they've decided to ignore the other two lobes. The joining of the ducts from each lobe forms the common hepatic duct. When the common hepatic goes past the cystic duct, those join together, and then you have what's called common bile duct. So down here it's common bile duct. Up here it's common hepatic duct. When the common hepatic joins with the cystic, that forms your common bile duct. This is all headed down. You can see kind of in shading behind the bile duct. The common bile duct will come down towards this papilla here, duodenopapilla. It's going to be joined by the pancreatic duct. The fluids from both ducts go into this dilated area called the ampulla if this sphincter relaxes. The sphincter is going to be controlled hormonally, and the hormones are coming from the duodenum. So the duodenum has control over whether it gets pancreatic secretions or it gets bile from the liver. The hormone is called cholecystokinin. It has a C here, and a K there, so usually they talk about it as CCK. They can call it cholecystokinin. It actually has a longer name you're going to see in your Silverthorne textbook, because it also is controlling pancreatic secretions. It has a pancreozymin or pancreatic enzyme connection. So in physiology textbooks, they call it cholecystokinin pancreozymin, which is a real mouthful. So once you said that, then you go back to calling it CCK, because that's easier. This sphincter here is called your hepatopancreatic sphincter. In Sherwood, still has the older term, the sphincter of Oddi, capital O-D-D-I, like that little dog in the comic strip, Oddi. I don't know if that Gary guy knows about this sphincter, and that's why he named his dog there, inside joke, ha-ha, or how he got that name. But anyway, O-D-D-I, same spelling as the little dog in the cartoon strip. The duodenopapilla is just a nipple-looking like structure that is in the inside of the duodenum. The gallbladder condenses bile and can store about 40 to 70 mLs of bile. You don't need a gallbladder, you can live without it. The fact that you have concentrated bile, and bile contains cholesterol and other things in it that can condense and form stones. You've probably heard of people having gallstones. If they're small enough, they can actually just go through the cystic duct and go up into the intestine and go out with the feces. But if they're too large to get through the cystic duct, and if you look, the cystic duct is in a straight tube. It spirals around. You can't actually see from the illustration that it's spirally arranged, but it kind of implies that with all these little step-like indentations of the wall of the duct. So the fact that it's spiraling down, you get larger gallstones stuck in that and in the duct, and that creates pressure building up and pain. So sometimes you can use ultrasound and break apart the smaller gallstones, and then they are cleared. But sometimes you get huge gallstones. They used to have a jar of gallstones in the lab, and they were about ½ an inch in diameter. They look like pebbles that you might find in the stream. They're just really solid. They're fragile, actually. You can smash them very easily, but I mean, well, if you abuse them, they fracture easily. But if they're in the gallbladder and they're that big, ultrasound's not going to clear them. So they go in there and they take the gallbladder out. Then your common hepatic, and common bile duct just dilate to hold the bile that's being made. And they serve as a reservoir. They're not going to concentrate the bile, but they can act as a storage for the bile. This picture shows you the orientation of the pancreas to the duodenum and the bile duct. There's also another little duct that goes off to the side here. Not everybody has two lobes to the pancreas. This is the main lobe, and some people only have that, and so they only have that main pancreatic duct. Other people have a smaller accessory lobe, and so they have an accessory duct as well. The duct has its own opening. When it exists, it has its own opening that's not at the hepatopancreatic ampulla. So pancreas is going to release a lot of enzymes that are going to help you with digestion of carbohydrates, fats, and proteins. I listed some here, just kind of general classifications, and sure we're going to get into the specifics. But lipases, which will break down fats, carbohydrates, which will help to break down the carbohydrates in your diet, the sugars and starches. Nucleases, because some people do eat meat and there are nucleic acids in the nuclei of the cells that are coming through the system. You want to be able to break those down and use some of those materials for your own use. And then proteins, proteases. The CCK, its job is to relax the hepatopancreatic sphincter and allow for these enzymes to have access to the duodenum. There's another hormone there that is also going to be released usually at the same time that the CCK is going out. Secretin is going to cause the ducts of the pancreas to produce and release bicarbonate. If you remember, stomach is right before the duodenum, and its pH is around 2, sometimes down at 1 when you are putting out a lot of acid. And that comes into your duodenum. The duodenum doesn't like that, and neither do these enzymes. They don't function at pH 1 or 2. They function in an alkaline pH, up around 8, 7 or 8. So you've got to take this acid that's coming from the stomach and get it up to pH 7 or 8. The duodenum will have glands that secrete bicarb, but then also the pancreas will release a lot of bicarb. And so from both sources, you kind of overpower the acid and get the pH up. So secretin, it's going to cause the pancreas to release the bicarb in hopes to get that pH up to 7 and 8. The cells here, those are your endocrine cells. We covered them last semester, but the hormones like insulin and glucagon, those are going, and pancreatic polypeptide, those are going to be involved in digestion. And also what happens with what the fate of the absorbed material is. So the duodenum is going to send some hormones to the pancreatic islets that will then cause insulin or glucagon release. So we've got to talk about those again, but we'll spend more time talking about what the these other cells, the majority of the pancreas is releasing, which are these enzymes. And then the ducts that release the secretin. When you look at a slide of the pancreas, most of what you see is this exocrine pancreas that's digestive related. So all this dark purple area, that is your pancreatic acini cells. And then duct cells will be creating the bicarb. This light area that's been outlined so you can see it easier, but if you squint and look at the slide, you can see this little patch of pale cells. That's a pancreatic islet, and that's your endocrine system. As you get older, things change, and you have problems with your GI system as well. Your stem cells that are down in the crypts, they become less able to undergo mitosis. So the surface epithelium is going to start to shrink and shrivel. If the surface epithelium, which is involved in absorption and digestion, is less abundant, then so is your absorption and digestion going to change. So you're less able to repair the cells that are up there on the surface of your small intestine. They last there about 2 days, and they're sloughed off and have to be replaced from below. So you have a high mitotic rate in your small intestine. But as you get older, that diminishes. And if your surface epithelium isn't there, then everything that's supporting it underneath, your villi start to get smaller, your plica start to disappear, and your intestine becomes flatter because it has less surface area. So everything just starts to go. The smooth muscle gets weaker, so more incontinence of the bowel. Back up to the stomach, you might get more contents of the stomach going into the duodenum than the duodenum can handle. So you might get an osmotic diuresis happening there. So you might get diarrhea, and that coming into the rectum, descending it very quickly, and less control of the sphincter. You got a lot of problems there. So depends for, that's a brand that I remember, but Poise and other things, huge market for the elderly. Even have ads on TV. You can get it in a plain brown wrapper, delivered to your door, so you don't have to go to the store and buy the stuff and be embarrassed. But the smooth muscle is also going to be moving things through the system. And if the smooth muscle isn't moving things through the system, especially in the colon, the material spends more time in the colon and more water gets reabsorbed than is supposed to get reabsorbed. So you get really compact, hard feces. I remember working at Kokini Daycare one day and helping somebody go to the bathroom. And they were in there about an hour and a half. And just grunting and groaning, and you had to keep your eye on them because when they're putting that much pressure down there, there's a chance they'll stroke out. So you gotta catch it fast if they have a stroke. But you wait and you wait and you wait, and what came out was about that big, and it was black like your t-shirt. It had been in that colon for a really long time. And that was all they got out, was just a little like walnut size, or maybe a little bit bigger than walnut. But that's all that came out. It had been in that colon for so long. It had gotten so dehydrated and compacted. That was the color. So constipation can increase. So usually what happens, you get constipated. Then watery stuff builds up behind. Clear the constipation, you got diarrhea. the water has come out, so you're dehydrated, that makes you more constipated. So you get constipation, diarrhea, lovely. Okay. Cumulative, everything, a little bit of damage, take good care of yourself now. What you can see, like up here in your oral cavity, take good care of your gums, floss daily, brush daily. Make sure that you clean the surface of your teeth. Flossing helps with keeping the gums healthy and active. Seems counterintuitive that you're pressing on the gums and that makes them healthier, but it does. Also, brushing them makes them better. If you lose your teeth, then your whole jaw and then your facial structure is going to change. So a lot of dentists now, instead of giving people dentures, if they're young enough and can stand the surgery, they'll implant teeth so that you have pressure on the bone due to chewing that will keep your jaw healthy, the bone healthy. But with loss of teeth, you have more problems with the other teeth in the area. Over time, you're going to have toxins accumulating, and that will create problems in healthy cells. You'll become less healthy. Cancer rates increase as you get older. Colon cancer goes up, so you need to have these lovely colonoscopies done. I think they usually tell you to start around 55 or 60. Pharyngeal cancer also increases, so you got problems up here, you got problems down there. Okay, take a break, and then we're going to start on Sherwood 16.